Kaustubh, let's start the session in a minute. So we'll start around like 7:04. Kindly be ready.
Sounds good.
Okay, I think we can go ahead. It's 7:04, around 7:04. And still, I think people are joining, so let them join as the session continues to. So first of all, I'm very excited and a warm welcome to all of you who have come, like joined from India as well as you guys, Europe. I think we have a lot of diverse attendees here. Thank you so much for joining this session. So myself, I'm Karthik, and I'm the co-founder and chapter lead of Neurotech X India. So NTX India is, as Neurotech X is an international neurotechnology community with a mission to advance the progress of neurotechnology worldwide. And in India, it's a very recently initiated chapter. We started around it's around 1 year old. We started in August 2022, and we've had some successful events in the past, including webinars where we have hosted Dr. David Eagleman, and we've hosted some professors from IITs. And yeah, and we have also had people like Ryan Field from Kernel, and now we have like Kaustubh, who has partially worked in Neuralink, and hope it's going to be very, very interesting. So at NTX India, we have a mission of advancing the progress of neurotech in India. And regarding that, we're trying to create this platform and bring in events, webinars to create awareness about neurotech, and also trying to integrate clinicians, professors, students, researchers in India to create a very vibrant and an ambitious ecosystem in India for the growth of neurotechnology. So with that, with no further delays, I'd like to introduce all of you to our guest speaker. I'm very excited to introduce Kaustubh. Kaustubh is a senior MEMS engineer and a neurotech enthusiast specializing in flexible neural probes. He began his research in chronic parylene probes at CMU and went on to work with leading neurotech companies such as NeuroNexus Technologies and, as we all know, Elon Musk's Neuralink, where he worked around 1 to 1.5 years, and where he worked basically on silicon and polyimide probes-based high bandwidth brain-computer interfaces. And his ambition, so after working there, he came back to India, and his ambition is to help people in Asia, from where he aims to work and learn with neurotechnology experts to help leapfrog this technology to the potential that it holds. I recently got an opportunity to talk to my friend Kaustubh, and we shared a lot of insights on the growth of neurotech, and he had a lot of interesting perspectives. And I thought definitely we should invite him to our NTX webinar. So with no further delays, yeah, thank you so much, Kaustubh, and we're excited to welcome you. The stage is yours.
Thank you, Karthik. I'm excited to be part of this mission by Neurotech X, and also just overwhelmed seeing so many potential neurotech experts and enthusiasts. I hope my presentation today excites you and then does imbibe some sort of enthusiasm towards neurotech, towards invasive BMI, and also just the efforts that we're putting in and how they can be clinically translated. With that, I'd like to start my presentation. We can usually I go for about 30 minutes, but if it gets a little tiring, don't hesitate to tell me to take a break, and then maybe we can just take a 2 or 3 minute respite. With that, let's begin. So my presentation today is going to be on intracortical neural probes. Thanks for the kind introduction, Karthik, but just recently my co-founder Professor Arjun Ramakrishnan at IIT Kanpur and I have started a company in India that manufactures invasive neural probes. Arjun's kind enough to be on this call as well, spending his Saturday evening with us. And we're just going to talk a little bit about the history of intracortical neural interfaces, iBMIs, and the potential applications for those, and including those in India and what the potential really is. And I'm just going to talk a little bit about my company and what we do and how our technology and the collective technology in the world can help better the state of neurotech today, and at the end of the day, help someone. So right now, okay, just to confirm, Karthik, you can see my screen, right?
Yes, Kaustubh, is it visible? It's visible.
Okay, great. So what I'm sharing with you right now is you'd think is a complex mesh of words, but in actuality, it's just a rat's brain. And a rat is one of the tiniest and most simple organisms, animals in this world to study. And despite that, if we are to figure out how a rat brain works, it's such a complex task because you can see how convoluted and complicated the wiring in its brains is. And really, if this is how complex a rat brain is, a human brain is going to be way more complex. So the point of neurotech today is we need to harness the potential of the brain, just like in any electrical appliance, which communicates in terms of electrical signals. Our brain communicates in terms of electrical signals as well. We use something called action potentials, which get fired, our neurons fire them, and then those get communicated through the brain, through the neurons, to the spine, and then distributed to the muscles to perform certain actions. And just like you'd fix an electrical appliance, to study, if you want to fix the brain and treat people with mental degenerative disorders, you need to figure out what the wiring is in the brain. And to do so, you need really tiny tools that can allow us to read and record the currents flowing in between the neurons and then the voltages at the terminals. So the mission today of the neurotech scenario is to build an intimate extension of ourselves. If we can harness the potential that our brain has and allow it to interface with a computer, we can truly be the very definition of an Android. It's no longer just a smartphone, but we and the smartphone can be a synergistic unit. So just in India today, mental degenerative disorders account for the second highest disease and death burden. And it's very unfortunate because there's not a lot of research that has happened in the past, primarily because there were lack of tools to study the brain. So just to give you a historical analogy, in the late 12th and 13th century, alchemy was quite popular. People believed that animals and humans worked on a mixture of chemicals, and that's what they thought the brain does too. Cut to the 16th or the 17th century, where the industrial era began and the gears were invented. People are thinking of the brain as having micro gears and chains and levers which tug and pull and allow certain actions to happen. It is only in the 19th and the 20th century that people figured out that this is an electrical phenomenon. But how do you measure it? The brain is just a tissue that's folded onto itself, and the neurons are so tiny, they're invisible to the naked eye. And that's primarily why the research hasn't taken off up until 50 years ago. Consequently, because we can't study the brain, we can't treat disorders like chronic pain, seizures, Parkinson's, epilepsy. But there have been certain clinical landmark medical technologies which have treated the symptoms, if not the disorder completely. Just today, for the government, the total economic burden to treat and to protect people with mental degenerative disorders is 68 billion rupees. That's a massive amount. That's a significant amount. And 35 million people in India. This was the stats from 2015, so I'm sure there are a lot more by now, but they've been living with neurological and psychiatric disorders. And it's unfortunate because these disorders can be treated. And it's just our mission to get there and help these people. So some of the landmark medical technologies that have been accepted widely are ECOGs for epilepsy, treating epilepsy using stimulation, deep brain stim for treating Parkinson's, EEGs and these can be used for treating or at least helping with depression, urinary incontinence. Cochlear implants have been popular since the 90s. Retinal implants these days, this company called Second Sight in California, which is commercialized retinal implants, allow people born with blindness to partially see images around. So, with the technologies that have already been accepted, us at A1 Neuro and people around the world who've been manufacturing invasive neural probes can help treat 15 million patients right away and just take their suffering away, allow their lifestyle condition to be much better. But where do we start? There has to be a starting point, right? And the single unit of the brain is a neuron. So we have to understand the brain and to understand the body and the functions that it performs. We have to start from deciphering a neuron to deciphering a group of neurons to a brain lobe to signals in the spinal cord and then eventually in the muscles. And those different signals that researchers today study, right from single units, which are emanated from neurons, to LFPs, which are superposition of different multi-unit activities, to alpha waves, delta waves, and EEG, and then finally EMG, which are electrical recordings from our muscles. just what I previously said, there's the different types of signals that can be recorded and that can be used to help treat a patient. And it depends from, it varies in terms of the invasiveness. So let's start with EEG, which uses surface recordings. It's above the scalp. And sometimes people shave off the hair, sometimes people tend to keep the hair, but it's fairly non-invasive. It's a little heavy and a little cumbersome, but still it gives us certain readings which are workable, which you can use to decipher a certain activity. But you can see from the signal on the right that it's very difficult to process these signals, and the spatial resolution is quite poor. So researchers then moved to something called as electrocorticography, or ECOGs. These are kept below the bone, so below the skull, and just on the surface of the brain. These have allowed us to understand the brain, even read information from brain lobes, and the signal processing for it is slightly better in terms of figuring out which part of the lobe does what. Finally, invasive probes, which are really tiny wires which can go in and record, just like a multimeter. They're able to record local field potentials and extracellular action potentials, or single units. And you can see from these signals, it allows us to account for better spatial and temporal resolution. So if you have an algorithm that uses some sort of filtering for a specific signal that's pertaining to an activity, you can now study cause and effect relationships in a brain and what a human does. And all of these require different tools. So EEGs require silver chloride electrodes, stainless steel dry electrodes. ECOGs again use different techniques for manufacturing. And then to make really invasive tiny neural probes, you need a technology called as MEMS, or thin film manufacturing, microfabrication. This technology was adopted from semiconductor manufacturing, developed by Intel and commercialized in the last 100 years, but it takes certain aspects and then it further builds on to make mechanical as well as electrical components on a silicon wafer. But to this day, this is the research that we've been doing, and it really shows how much we've been able to, or how vast of a problem we're trying to figure out, we're trying to solve. And to do so, I think we need the right set of tools, because there are ample amount of researchers working on this. And just like the Human Genome Project, which started in the early 90s, there's the Obama administration announced the Human Connectome Project, which will allow researchers across the world to collect data and to understand how, or just map the human brain. To delve into the history of neural probes a little, this is just to give you a perspective and how this field even ventured into what it is today. So we started off with, what is a multimeter? At the end of the day, it's just a single wire. So you can put a cloth hanger in the brain and still record activity. Probably not the best idea, but the whole effort in the last 20 years was to miniaturize a microwire and have as many microwires and as small as the footprint as possible. So from history, this is collected by Jamie Hedke at Nexus Technologies, but they started off with making silicon arrays, because silicon was what was used for semiconductor manufacturing, and it was a simple substrate material to use with. So in 1996, they made multi-shank electrode implants. There were retinal implants. This was the early work that was done by researchers who were associated with Second Sight, and then this technology was eventually commercialized to make retinal implants. But you can see that some of the dimensions on here, the electrodes, are fairly large. And such large features the research was to make them smaller and miniaturize them smaller, so that you could get more spatial resolution and record from as many neurons as possible. And another cool research was from Simon Fraser University in the mid-90s. This was to record from peripheral nerves, where you had a hook for a researcher to latch onto, and with a needle poke the peripheral nerves and record activity from them. And a lot of the work was done on silicon, but unfortunately, researchers realized that silicon does not have as good of a clinical translation as other materials would, because silicon is brittle and it's toxic to the human body. So the research from the mid-90s then pivoted to give way for organic polymers or alternate materials, which are not just biocompatible, but biostable as well. But talking about BMIs and not just electrodes, in 1969, this was a pioneering study by a famous professor named Fetz. He's still working, so you can tell how dedicated he is to his work. But in the 1969 paper by Eberhard Fetz, he managed to figure out that monkeys could voluntarily control the firing rates in their brain when they were awarded with food. And with that, he realized that if a monkey can control its firing rates and can control the neural pace, humans could too. So in degenerative disorders where certain pathways, neural pathways, degenerate or don't work or fire too much, there is a possibility for human biology to avert those pathways and make new pathways, consequently helping treat some of the diseases that are out there. And even with some medical device intervention, this is absolutely possible. And this was one of the pioneering research that happened in 1969, which paved way to invasive BMIs becoming a reality. And isn't it really cool? Like imagine, as a child, I always tried to, I was told that you only use 20% of your total brain power. And I thought, if maybe I have control over it, and I used to shut my eyes and think hard. Didn't succeed. But seems like Professor Fritz did. And then very exciting work that eventually ensued. Cut to 2006, where BrainGate, this company born out of Brown University, was able to pioneer technology where they actually implanted an electrode interface, coupled it with a decoder to understand the neural signals, and then have some sort of a sensory feedback and output mechanism, the output mechanism being the robotic arm. And it's amazing to see because this image is part of history today. This lady with the Utah array, these are single shank electrodes made, we call them the bed of nails, but these are made on a silicon substrate. These are implanted in this lady. And the researchers at Brown were able to control a robotic arm for her to control and feed her. Just eventually, it was just a sip of water, but this was pioneering work, and this made history. Couple of years down the line, universities such as University of Pittsburgh, University of California at San Francisco, they did these human studies too, and they eventually found out that you could push more and you could put these devices into other areas in the brain and get good results. So eventually, the whole idea is to improve the condition of living and also provide therapy to people suffering. This was a recent study on the bottom right, the image that you see on the bottom right, was a recent study where two individuals with tetraplegia, so their arms and legs are paralyzed, they were able to communicate with just their thoughts on WhatsApp. And that is amazing. Even as an able-bodied person, if I could do that, that would be remarkable. And this just lets you appreciate how much this field has grown and how much potential there is in this field. But right now, we're still limited to the primary motor cortex. So the primary motor cortex is an area in the brain that allows motion control, and it's studied fairly well. So a lot of researchers continue to push research in that direction. But there are other regions of the brain, deeper regions of the brain, which can help with more therapeutic applications. Oh, I'm seeing chats here. Karthik, are there questions coming in or?
No.
Okay. All right, cool.
I think
Cool.
the audience can directly switch on their video and audio and can ask you directly.
Awesome. So if anyone does have questions, feel free to interrupt. This is an informal conversation, and I'd be happy to answer any questions. All right, cut to new electrodes that are seen today. These are some of the images and some of the companies that are working on cool solutions, unique solutions, and not just silicon microwires. So on the top left, you can see a polyimide array. This is an ECOG array that was made by University of Wisconsin at Madison. And they were able to use these flexible polymers and biocompatible and non-cytotoxic polymers to enable chronic recordings from a monkey. And this is quite a pivotal study because since 1998, there's been a lot of work done on polyimide. And both Arjun and I today are furthering this work by making our own polyimide, but I'll get to that part later. Another solution so the unique part about this solution was they were able to put multiple electrodes on a single shank. Now it's important to do so because when you insertion, when you do an implantation, you don't want to do too many surgeries because that causes trauma to the body. And there's always neuronal death around the site of implantation. So if you have multiple electrodes present on a single shank, you're able to now figure out more activity from a smaller surgical footprint. And that's a disadvantage of the second electrode strategy, which was pioneered at University of Utah and now commercialized by a company called Blackrock Microsystems. These are essentially a bed of silicon nails with iridium oxide as their electrode material. They are, I'd say, pushed into the brain, there is some biological damage, but these have shown excellent promise in terms of chronic lifetime, and these are the ones that were implanted in humans. So here the disadvantage is that they have a single electrode per shank. So in order for them to increase the area from where they record in the brain, they have to make more and more needle, and that sets not just a processing challenge, but even a biological challenge, because you're now facing trauma. And consequently, while this is still an FDA-approved technology, work on this has, at least the delta on the progress that's been made on Utah arrays has been minimal in the past few years. This then led way to CMOS technologies, where people figured that if we have to miniaturize these recording electrodes, why not just use the MOSFET technology that's available in semiconductors and use the gate of that MOSFET to record neural activity. And this is promising, but again, the limiting is that clinically translating this device is tricky, but it's still made on silicon. However, for academic purposes, this allows for a higher channel count, but it's still fairly new, and the adoption of this technology is the jury's still out on that. The next image here is that of an optoprobe. An optoprobe is another cool technology pioneered by Carl Distroth and Ed Boyden at MIT. They figured out that if you inject some cells, an animal cell with a virus called channel rhodopsin, you can allow optical modulation to record and stimulate from the brain. And so researchers then, while this was a pioneering technology, it came very close to winning the Nobel Prize. And researchers then, engineers, I would say, then moved on to making devices which could do not just electrical stimulation, but light stimulation as well. The image on the bottom left is that of an ECOG, and you can see that there's been efforts to make different patterns, use different substrates, and miniaturize the ECOG, because this is also a clinically translated technology, and it's quite widely used for epilepsy treatment. These two images are those of multimodal probes. So this is another CMOS probe, and then this is one with fluidic connections. So neuropharmacology is a huge field, and if there is a pharmaceutical treatment that is possible for mental disorders, then a device like this would permit for it. So quite promising. And then up until recently, I'd like to think that the Neurotech wars have recently begun. So many companies have come up. One of the companies that I worked at recently, Neuralink, Elon Musk led this team, and the whole effort was to miniaturize not just the electrodes but also the packages. And this technology, the N1, they're calling it the N1 device, pushed for even thinner and even smaller devices that would be implanted in the human brain. And the pack for this was so tiny that you could just leave it in the head. Typically, the package is kept outside the head next to the collarbone, but with the Neuralink device, it is so tiny and so compact and allows for fast wireless communication that you can just keep it in the skull, and it works perfectly fine. So the study is done with pigs and monkeys on this. I had the privilege for they had two demos, and I had the privilege for making both the implants that were showcased in the animals and the demos. So very, very rewarding and creatively satisfying work. Finally, on the bottom right, this is again a device that's recently gotten an FDA 510K approval called NeuroPace, where they use ECOGs for recording and then invasive depth probes for stimulation. So it's a closed loop feedback system which helps people with Parkinson's and epilepsy. The image on the bottom here is that of a Electrode. This is pioneered by Synchron, an Australian company, and Dr. Thomas Oxley. He figured that instead of entering the brain directly, why not enter the brain through veins? And he figured that if we have a catheter-like device with large electrodes on them, you can put them in for a very long time without having too invasive of a surgery. Just as of September 2020, they reported some preclinical trials, very promising, and this company is able to raise close to $50 million in funding for high volume production of this device. The image on the bottom right is that pioneered by Dr. Charles Lieber from Harvard. It's that of this device is called the neural lace, and his idea was to inject this mesh into the brain and kind of let it assimilate with the neurons, and consequently you would always have some sort of recording activity. Unfortunately, the work on this has recently stopped, but I'm sure someone would pick it up if they show more promise in vivo. Now, cut to 2018 and 2019. Again, I get very excited when I see this research, but people wanted to now make smaller devices, right? You don't want to make probes that are wired, that are tethered. You want to make wireless devices. And there were 4 companies that did this. One was Neurotech by iMac in Europe. They made these CMOS devices again, which could be wirelessly recorded from. These are still at the animal trial stage. So the jury is still out on them. This technology on the right, NeuroBrain, was pioneered by Brown University, where they made minute CMOS chips that could be distributed inside the brain, like a network, and they could communicate with each other using Bluetooth and moved around using ultrasound. Very bold and very innovative technology. And again, I'm very excited to see this. But one of the, at least in the last 25 years, this work has been pioneering. And if they do get good results, I'm sure we're not too far away from seeing the age of the Android. And the image on the bottom left is that of a version of NeuroBrain that was developed by UC Berkeley prior to the NeuroBrain. It's called the Neural Dust, and they've then ventured into then commercialized this technology through a company called IOTA. Similar idea that you could make tiny grain-like chips and which could distribute themselves across the brain and then communicate with ultrasound. And finally, very recently, the Synapse probe from Italy has come up. The difference between Synapse and NeuroPixel, they're both CMOS probes, but the difference was the Synapse recorded more channels at any point of time. So NeuroPixels, because of the technology, limits them to recording only from 400 channels at a time. But with the Synapse probe, they could record from 4,000. So that's an order of magnitude higher. And it's a scalable technology. So excellent work again. A lot of researchers have flocked to them to try this out, but the jury's still out then. But this research is as of 2 years ago. So really cutting edge, indeed cutting edge. And that brings me to what me and what Arjun and I have been doing in India. So we figured that the US markets are fairly saturated now. There's a lot of proliferation of these neural devices, but Asia is still not. There still hasn't been a lot of research done in Asia, and we figured that together we want to do something, and together we want to help people in Asia, starting with India. So we've developed intracortical probes made with polyimide. But this is a proprietary polyimide that we've put years into, where the chronic lifetime of this polyimide is much longer than what's commercially out there. I'm quite excited for it because this gets us closer and closer to clinical translation. We use state-of-the-art thin film MEMS technology in India. We've developed it, and we can go down to 4 microns in thickness, potentially being one of the thinnest probes out there in the world. And this, because we have our own polyimide, it lends itself well to this technology. So what I'm showing you on the left is an image of how it's fabricated. It's done on a silicon wafer, and you can put different designs that researchers customize on it at a time, and then release them off the silicon wafer for use. But the cool thing about these devices is these are 100 times thinner than our hair. So you imagine how thin and flimsy they are and how difficult it would be to insert them. I'm talking about the thickness of these devices. This part is what goes in, not the ones with the rectangles, but the ability to make not just flexible, but ultra flexible devices is what got us excited. And we developed this technology at Ava Neuro. Just recently, we've tested them for cytotoxicity. We've done some animal experiments with them. I'd be happy to share them more towards the end of the presentation. Now, I keep talking about these electrodes, and I keep talking about different companies and their designs, but really, there is no one answer. There isn't a solution for at least when we're studying the brain. A lot of the neurons are different. The regions of the brain that you want to target are different. So there isn't a one solution fits all strategy that can be adopted here. And that's why with thin film MEMS technology, you can now change the length, the width, the dimensions of the probes. You can change how the probe looks. You can base it for how you want to surgically insert it. Depending on the researcher, there could be innovative ways to do surgery to reduce biological damage. So this thin film technology essentially allows us to make customized designs, which is really rule 101 of this game. That if you're able to customize your design, you're off to the right start. So what we at Ava believe is if you can draw it, we can make it. It is also NeuroNexus, another company in Michigan. They do this too. But this technology is so ubiquitous and exciting that it lends itself very well to for researchers to study the human brain. And I know I'm showing you images, but it's very hard to understand the depth or the thickness of these probes, how it translates to the brain. And the image on the left is I've got that from the link and one video that was implanted in Pager the monkey. And you can see that the devices are tiny enough to avoid the blood vessels in the brain, let alone harm or hurt the brain. So with this, with such a thin accessible technology, you can now put a lot of tiny arrays without causing trauma to the animal or human. And this is exciting work, and it just puts into perspective how thin these devices are and how difficult they are to insert. A lot of neurosurgeons have come up with great ways to insert them and reduce biological damage, and that's kind of pushed how we can study and the amount of data we can gather from neuronal recordings. This image on the right is that of a recent experiment just as of last week that our probe did in a hawk moth. So hawk moth, really tiny animal, and our probes were able to be put inside the neck muscles of the hawk moth and record some EMGs from them. Another cool thing that I want to highlight with flexible materials as opposed to silicon is also correction, silicon and not silicone. Don't confuse that. So this image on the bottom right is an explanted probe, and you can see that there's no damage to the probe, there's no damage to the animal, and this probe be implanted and explanted as many number of times. So in an actual realization in a clinical setting, if something does go wrong during the surgery, this probe can be explanted easily without hurting or damaging the brain. So things like getting upgrades maybe for software or for hardware are really possible because this process is, you could do it over again. Now, just to this conversation, today's talk was more about introducing iBMIs and how we use thin film MEMS technology to manufacture these. So I'm just going to take you through the process of how generally these devices are manufactured and how at Elon Neuro we do them. So initially, our surgical and neuroscience team sits with the customers, the researchers, to understand their animal model and then figure out a way for them to insert these devices and record good signals. So things like electrode area, the width of the probe, the length of the probe, how flexible they want it are topics of conversation. And once we figured out a good design, we translate it to a CAD model. So this is then made on a CAD software and then put onto something called as a photo mask, which then used to, it's something like photography where you're exposing this layer onto a silicon wafer and then slowly stacking thin film layers on the top. This design then gets put on a mask, and you can see the image on the bottom right shows you a photo mask. It's a quartz plate on one side and a chrome plate on the other, and then there are openings on the chrome plate which align to the designs that you've made, and then we perform photolithography on it for manufacturing. So to take you through the manufacturing process, initially we start with regular silicon wafers, we clean them, we descum them, and then we spin a layer of our spin coat a layer of our polyimide. It's proprietary, and there's a way we do it. It's different from what's traditionally done out there. And then we do lithography. So with lithography, we're able to, if you see the image on the left, this is how we do patterning. So when we draw out the CAD model, we then utilize we spin coat a photosensitive polymer on a wafer and then expose it through a mask. So with this, you're able to put designs and then have thin film designs be stacked onto the wafer. This is an example of liftoff where you could put down metal and put down thin film traces, which are used for traces and recording electrical data. The next step is then to spin coat a second layer of polyimide, which acts as a second layer of insulation on the top. And then we use oxygen plasma etching to open electrodes. So the electrodes are essentially open on the top part of the polyimide, and then the outline is etched off to singulate the devices. So on the bottom right, you see the devices on the wafers being singulated, and then on the top bottom left, sorry, and on the top right, you can see the electrodes. So these are platinum electrodes with thin film ti-platinum gold traces that so you're not just able to utilize and harness the benefits of using precious metals, but with this, you're also able to drop the cost significantly because you're not using a lot of metal. Finally, these devices are then released off the silicon wafer in IPA or in microscope, and then they're assembled onto PCBs to connect to amplifiers and recording stations. So just to explain, I know I just used the concept of lithography a little bit. I'm not going to it just in a little more detail to explain how it works. So the idea is similar to photography, where you have a photosensitive layer which you expose using a reticle, and then the exposed region on the resist is then developed, so it's dissolved away, and that makes openings. So when you have that, you're able to either put down, deposit material, or you're able to etch material off. So an image, and there are different ways to do it. There's UV lithography, there's deep UV lithography. Recently, there's what Intel uses is extreme UV lithography, which goes down to 10 nanometers and smaller. But we use contact lithography, which allows the electrodes which allows the critical dimensions to go down to 1 micron. So the images here show you what a photomask is. And then this is called the mask aligner, contact mask aligner, which allows you to align features on the wafer to the next layer. And then with a UV source, you expose it. Just an image of how the wafer and the reticle align. And then finally, the photoresist layer gets developed to make features on it. So eventually, you're finished with either putting down metal or taking away metal. You can then dissolve this photosensitive layer away, and then just have the metal or the etched portions remaining. Now, two ways you can proceed after doing lithography. You can either put down metal with deposition. They can be either physical vapor deposition or chemical vapor deposition. What we use is e-beam lithography, and I'm just going to tease it a little bit. But it's a system where the machine is in high vacuum. It goes down to 10⁻⁻⁶ torr. At this stage, you have an electron beam that gets shown onto a metal crucible. This is just a tungsten crucible with metal pellets in it. And the e-beam, the heat from the e-beam is able to not just melt the metal pellets, but also evaporate it. This evaporated metal then goes and then evaporates and condenses onto a vase. And that's how we pattern metal traces down. So we do this with titanium, gold, and platinum. And then eventually, once the deposition is done, we strip off the resist. And just have the metal remaining in the areas where we want it to be. Another technique, and again, this is a cool technique. We're using etching to do so. Now, the challenge with etching polyimide is polyimide and photoresist etch at the same rate in oxygen plasma. So if you have, let's say, 10 microns of polyimide, you need 10 microns or more of photoresist. Otherwise, it's just going to etch through the photoresist and etch into the polyimide. So what we do is we're using something called as a hard mask, where we put down another sacrificial layer, aluminum in this case, and then pattern the aluminum to make openings. So instead of having a photosensitive mask, you now have a metal mask, and then we proceed with the oxygen plasma etching. Beautiful color, right? Like it's a nice purple color that you see in the oxygen plasma. And this over time etch the polyimide to form volatile compounds, which then get sucked in, neutralized, and then released. So very cool technology. It's fairly recent, just made in the last 20 years. NASA was the one to pioneer it, and I moved. This was polyimide then became a popular choice to use in organic flexible electronics and also in high temperature electronics. Now, talking a little bit about invasive BMIs and how they work. So far, the lowest hanging fruit, or the easiest way to translate a BMI, is to put it in a motor cortex. The motor cortex, primary motor cortex in the brain, is an area in the brain that sends signals to control your limbs, to control motor actions. Now, oftentimes people with these diseases, with mental degenerative disorders, have spinal cord issues. Either they've been in an accident or they've been born with a defect. But the fortunate thing is that because the spinal, if the parts of the spinal cord are not working, that doesn't mean the brain is not working. So if we pay into the signals that are created in the brain, we can then meander around the spinal cord and just use that to actuate something else, like an exoskeleton or even an arm, a prosthetic arm that's connected to your body. So if the whole point of doing an invasive BMI is to leverage those signals and those signals down to a single neuron, so you get very high spatial resolution. Now, someone, I posted this on LinkedIn the other day, and someone commented saying, "This is rubbish. Like, why would we put something in our brain? It's dangerous. We could do this with an EEG." Very true. You could do this with an EEG, but the correctness of that technology would drop significantly. And it's, when I say significantly, it's orders of magnitude different, because while EEG works great for certain applications, doing motor control like this is very difficult with EEG, especially with so many degrees of freedom. And that's why iBMIs are there to stay. They're not going out of business. So the basic components of a BMI are the electrode, which records electrical activity from the brain, and then that data goes to a decoder where it gets filtered, it gets amplified, and then it gets sorted. So something called spike sorting, which means you collect activity, electrical activity from the neurons, and then you bandpass filter it for certain frequencies of interest, and then you time map it so that you figure out what the cause and effect relationship of an action is. Now, with that, there is an output device. It could be an actuator, it could be an exoskeleton, it could just be a TV screen, which allows you to use those signals in the brain to perform actions. And then there is feedback back to the person. So whenever they see, just what the FEDS paper predicted, where the monkey was able to control the firing rate of the neurons, if a person with spinal cord injury is able to see a computer monitor and uses brain signals to control the cursor, he can then use his own biology to control it better, to get better at this technology. And that's why it's while this is difficult to do with animals because it's hard to communicate the feedback to animals, we researchers and scientists do expect that this would be much easier to do in humans if this gets proven to be safe and a safe technology for use. Now, on the bottom right, again, an image from Brown where two people use this to control cursor for texting, and then this language texting was then used to communicate through WhatsApp. So very cool technology. Professor Ramakrishnan was on this call and my co-founder. He works a lot with the decision-making part of the brain, and he develops decoding algorithms to study those. Again, to talk about decoders a little, the three components, very basic. I'm sure there's more to it. I'm not an expert on it, but from my understanding, I'd love to tell you something. So the signals that come through, they're initially filtered and amplified to be able to detect certain shapes. Now, threshold filtering is a fairly common technique where they just look at the amplitude of the spikes recorded, and if that amplitude goes beyond a certain threshold, you detect that as an activity. And then this activity is then discretized, where you have individual components of discrete data, and they get clubbed together to detect certain cause and effect relationships. This processing occurs, it sounds trivial when I say it, but it's a lot more complicated to do it in person. And there has to be a lot of training and machine learning that also goes on to figure out the actions of the brain, because each day the brain, there are about 85 trillion synapses that happen in the human brain. And to figure those out, it's difficult to do this with a trivial algorithm. And now this is the exciting one. This is the second part of the call. I'm just gonna maybe ask Karthik if people are tired or they'd like a break, and maybe we could take a break for 2 minutes, just get some respite, and then I'll finish off with the rest of the slides.
I think if so, we can just audience can just put it in the chat if you need a break or so.
Yeah.
Kaustubh, are you gonna what's the time for the second session? Is it going to be like 15 minutes or?
Yeah. Okay, sounds good. So
okay,
we could just begin.
People are enjoying, so yeah.
Okay, thank you, Deepak. Very kind of you. All right, awesome. So researchers realized that. Pardon me, I'm just going to get a sip of water. Great. So researchers realized that we're not stopping there, right? We're not just stopping with decoding. We have to go beyond. We're not just making a diagnostic device, but to make a therapeutic device. So we figured a lot of research came out of UT Dallas. This Bitan Chakravarti who's on this call. He worked with Dr. Kogan, who wrote a pioneering paper on cortical stimulation. But there are materials that use either capacity or Faraday charge transfer to inject current into the brain. Now, what happens is a brain is just like an electrical circuit, right? Now you've got a stream of charge passing through the brain and then into the muscle. Now, imagine if you're able to, let's say you've got a seizure where the brain is passing too much current, right? And that's why your arm moves or your body moves and you get a fit. But if you want to stop that, there is a potential for using external charge with these devices, which gets injected into the brain and inhibits any charge transfer to go through. Now, there are two mechanisms by which this can happen, either inhibition or excitation. So in areas where you want certain actions to happen, like let's say someone's having difficulty moving their arm, then you could use cortical stimulation to inject charge into the brain and then cause a lot of movement. And the other side to it is you could inject charge in certain other areas to inhibit charge firing. And this technology is not possible. Again, like that comes with an asterisk because the jury's still out there, but it's not possible with surface stimulation or EEGs or other non-invasive technologies, because you're just missing out on that kind of spatial and temporal resolution. The brain fires in like milliseconds, so it's difficult to stimulate something with that high of a frequency and that high of a charge non-invasively. And that's my work continues to be in this field. Now, these were electrodes on the bottom left that I designed in 2018. They used the novel high-K dielectric material for capacitive stimulation. And this material, again, with stimulation, you could also do, similar to stimulation, you could also do voltammetry. So in synapses where there are neurotransmitters used for chemical, the neurotransmitters are chemicals in the brain that essentially allow charge to transfer, information to transfer between two neurons, the axon terminal and the axon. So you can use cyclic voltammetry to detect those chemicals as well. So a lot of, when you hear this research saying scientists figured out that serotonin helps with sleeping, or norepinephrine works with reducing heart attacks. I'm making this up, don't quote me on this, but they were able to figure this out because they use these probes with cyclic voltammetry to detect and neurotransmitters. And you can also code these probes with certain proteins, which again, like this is beyond the scope of today, but I'm excited, so I'm sharing this with you. But there's a male to female part essentially of these proteins, and when you detect those neurotransmitters, you're able to understand their role better. So cortical stimulation goes the end game. I think that's where people will really get help, and that's where the community and the market will see money, because people would buy this. If God forbid, but years down the line, if I get Parkinson's, I think I want something like this, because there's a technology called deep brain stimulation where they're large electrodes and they're not too targeted, but when you put those in the brain and then when you inject a lot of charge, you're able to reduce tremors, and this helps with people who suffer from SM tremors. So it's a wonderful technology. It's not completely understood as of 2021. There's a lot of research going on in it, but there is a high potential for something like this to happen. And oftentimes, I don't know if it's coincidental or not, but science always follows fiction. In the movie Avatar or Avatar, if you're from India, I don't know if you remember, but the avatars are able to communicate with their land, with their tree, using certain indices, right? And it helps them heal, it helps them work, and really it won't be too difficult, or maybe it's a little audacious, but it is a possibility. It is a possibility with humans, and this technology is ubiquitous, so it can help with a lot of diseases, not just the ones we know of. Right. So today, where does the competitive landscape start, and what are the streams of thought, right? So there are 5 different streams of thought and what researchers are pursuing. So the first one, very primitive ones, the silicon arrays. These are still very common, highly used in research because traditionally they've worked well and the infrastructure surrounding those probes do exist. You then have, these are multiple electrodes on single shanks, shank meaning a certain one particular needle, and they have a smaller footprint per electrode. The Utah MEAs, again, these are chronic devices. Oh, the silicon arrays suffer from chronic use. So it's difficult for us to use them in chronic settings because they fail in a few months. The Utah MEAs work really well. They lend themselves well for chronic use, but they're limited by the resolution by the scalability that they can achieve. CMOS probes, again, a new alternative, but clean translation is difficult because they're still made on silicon. And then the silicone arrays, which is again pioneered in Germany. They're made from silicone. Silicone is the food grade material that you use for your food boxes or ice trays, your cake molds, and that's a very inert material as well. But processing with it is difficult, and you can't spin thin films of it. So scaling it down further is challenging, and that's why at Ava Neuro, Arjun and I developed our own ultra flexible polyimide arrays because the buck doesn't stop at 10 microns. It has to go thinner. It has to be smaller. And the belief is that the smaller you go, the less the brain identifies this as a potential for an object or potential for harm, and the less response or biological response it sees, allowing it to be used for chronic settings. So very versatile technology, and this is scalable. So right now, with what we have, we're able to scale this down to 4 microns. And just from the information on the slides, you can tell that that's an order of magnitude difference, right, from what's out there. We're close to the silicon arrays, but that movement from 15 microns and below, that's difficult, and we've got that with our technology. This is a stupid picture because the 1 rupee coin keeps changing. I just had it in my stock photos, but you can probably relate the size of these probes to or how tiny they are. But if I were to tell you that they're less than 100 microns, so extremely thin, extremely tiny, and with multiple channels, multiple electrodes on them. The competitive landscape, it's filling up quickly. There's a lot of work being done in the US. Almost 94% of the world's neuroscience is being done in the US. A lot of it is being done in Europe now, but Asia is a growing market. Asia and Africa are a growing market, and both Arjun and I are proud to announce that we're the first neurotech company in Asia, in South Asia. There are a few other companies who are working on it, but they still don't have a product yet. So we're proud to hit the market, at least in India first, and then help make a difference in this field in India. And so far, like, because the US has been a big market player, it's been difficult for Indian researchers to import these devices or use these in hospitals. And from the preliminary work that we've done, we've got some great testimonials that our devices would allow for an easier integration of the electronics industry, the medical manufacturing industry with hospitals and researchers, and it also brings this down. India's, again, there's a massive amount of data and so much research that's happening that together, like if we collect a biobank, I don't think possibly decoding the brain would be too far out. Maybe a decade and a half, but it's a realistic prospect, and we're excited for this. So promising results. And then finally, I want to you guys know me already, but I also wanted to introduce Arjun, who's part of AVA. He's a professor at the Bioengineering Department at IIT Kanpur, and he's also working on some EEG and invasive startups. He worked at UPenn and Duke before. In fact, he's worked with some of the early founding members at Neuralink, so we had a lot in common with them. And finally, this brings me to the end of my presentation. I just want to end with a high remark saying, the work's been done in neural probes, the silicon neural probes or Utah probes. They made an impact. They made a change. And then in at least the last 5 years or so, the CMOS arrays have made a difference. But now with us, with Ava Neuro, and then all the participants on this call, I think we're the next wave. We're the follow-up wave. and we want to be there at the right place at the right time and the right capacity. And with that, I welcome you all to the second machine age. Let's get into the age of the Android, and let's bring about the next evolution. Thank you so much for your time. The floor is open for the questions now.
Yeah, thank you so much, Kaustubh. Yeah, maybe we'll open the questions, and if the audience has any questions, you can just unmute yourself and ask your questions directly. I think there's already a question by Vaidehi. Do you want to unmute yourself? I think. Kaustubh, are you able to see the question? I'll read the question for you.
Yeah.
Can polyimide probes be used as a potential tool for therapeutic targeted drug delivery of peptides or siRNAs, which are primarily hydrophilic and cannot cross the blood-brain barrier with conventional delivery systems?
Yes, they can. There's been a lot of research done at University of Pittsburgh. There's a researcher, who's coated these probes with serotonin and other proteins and hydrogels, which can allow, and with microfluidic capabilities, they're allowed to inject pharmaceuticals inside the brain and then treat them not just electrically, but chemically as well. So definitely, this technology lends itself to that possibility.
Great. Okay. I think Bitin has another question. I think he has already sent a question. So his question is, to implant these devices, Neuralink uses a robot to perform the surgery. The precision is therefore very high and can easily penetrate the PR mat of the brain. So what's your strategy?
That's a good question, right? So the robot that Neuralink uses is picked up by a needle syringe, which is thin, which is 40 to 50 microns wide. So we use a similar strategy. We either stiffen these probes with a temporary stiffener that allows them to be strong while inserting, but they're water soluble. So the moment they hit the cerebral spinal fluid in the brain, it dissolves. And the other strategy that we use is to thread a microneedle through a loop in these devices and then put them in, and then put the microneedle out, leaving the probes in.
Okay. Audience, if you have any questions, kindly unmute yourself and ask yourself. You can ask the question yourself.
Hello, Kaustubh. It was a very nice talk.
Thank you.
I just wanted to know, how much you have conducted this on mice, or what animals you have used this probe?
We do a lot of animals, small animals, large animals. So my co-founder Arjun, he works with monkeys. So these probes can be used in monkeys. The only difference is the designs are different, but we've done them on rodents and hawk moths, basically really tiny. They're the size of, they're somewhat larger than a fruit fly and then smaller than a grasshopper or a fly maybe. Yeah.
Okay. And like in monkeys, how much time duration you have kept it, like maximum time duration what we have used for, like how was the degradation of signal over the period of time?
That's a good question. I'll probably get into it in just a little later. Maybe it'll take a minute to explain. But the moment you put the probe in, it incites something called as inflammation. Of course, it causes inflammation because of engine, and it incites something called as the chronic body response. So the initial response, just like when you prick your finger with a sharp object, initially there's blood flow and there's injury, so there's inflammation. The area around it goes red.
I am a doctor, just to know.
Oh, I see. Okay.
I'm a neurosurgeon.
Nice. I'm not, so in terms of for you, like we get gliosis essentially. So you get a glial response, which for people who are not from the medical field, glial cells are essentially an umbrella term used for cells that do certain functions in the brain. So what happens when there's an injury is that these cells come and wrap around the probe. It's believed that it's like, say, a bacteria that goes in. The body's way to fight it is to wrap these around with these glial cells and suffocate it to death. Suffocate in terms of both like food and others, but the same process happens to neural probes, but because these cells are quite thin, you can still record. So initially, when you put probes in, an impedance is a good indication of the recording performance. So the impedance goes high initially. And then once the gliosis settles down, once the body thinks that the glial cells have coated these probes uniformly, then the impedance plateaus. And that's at that stage where you start getting your chronic recordings. So the probes by themselves operate, you get useful data for quite a long time. With these, the best research that's happened on these polyimide probes in a case where they lasted for 2 years, and then the researchers stopped that experiment because statistically a lot of the arrays failed. But when you, it's a combination, it's a permutations and combinations game. If you have a wider electrode area or a bigger device, then it lasts longer. But that's not the purpose. We need to go smaller and thinner and more electrodes so that we don't cause a lot of gliosis. And at the same, we ensure that the recording performance is good too. So for Bhushan, the moment these probes go in, it incites gliosis, and then until that gliosis becomes unsophisticated, that it's around the 14-day mark. That's when we start settling down and getting the chronic performance.
Okay, fine. And just means out of the very question, how much time you are expecting it to start your human trials?
Oh man, I wish I could do it tomorrow, right? I wish I could do it on me. I trust my technology. Uh, so I don't know. I think like there's a lot of bureaucracy and like not in the bad sense, in a good sense, because like you don't want to kill someone, right? Just like with the COVID vaccine, even after going through speedy trials, it took them almost a year to come up with, to get approval. So something like this, unless they're stable for a year, 2 years, and they don't see long-term damage, it's challenging. But a lot of companies have applied for an FDA 510K, which is pre-market trial. So pre-market trial work with all cohorts of patients, and then you study them. So there is potential for translation. It's just a matter of like funding. It's a matter of getting patients to willingly accept this. It's also there are ethical concerns that come with this too. So all the researchers across the world are slowly like figuring that phase out. But we're hoping Neuralink leads the way. And then
no, but I guess if you want to apply in India now, that you have to apply for DCGI directly, correct?
Apply for, sorry, DC?
DCGI, the Indian FDA, like.
Yeah, that's true. That's true.
If you're planning to do it in India, like really, I want to be a part of it. Yeah.
Oh yeah, we'd love to. I mean, feel free to shoot us an email, and then I'm sure we can talk. Always excited to collaborate with people. More the merrier.
Sure. Yeah.
Thanks, Bhushan.
Hi, Kaustubh and Arjun. This is Bhupendra, and thank you very much for this. Was a fantastic presentation, and in fact, fantastic work you guys have done so far. I'm wondering, I mean, if you don't mind, would you like to share some price range what you have within for these props you have developed?
Yeah, that's a good question. So it really depends. If I go back to my presentation, right? Oops. Oh, there we go. So we manufacture these on silicon wafers, right? And then for us, we price the real estate of this silicon wafer. Where someone wants a massive probe, then they pay more, and someone wants a smaller probe like this, they pay less. So it's really the price of the real silicon real estate and not the probe by itself. But ordinarily, I would probably think of it as in tens of thousands. So not more than 1 lakh and not less than maybe like 15,000.
So that is INR, right?
INR, yeah. Oh boy, like not dollars. Who's gonna pay like $100,000?
So by the way, what is the data? What is the data collection process? I mean, is there something you're giving some interface? So your part is just actually helping us to give this kind of probes develops, right? And then you're giving some sort of interface for data collection.
Yes.
And then after it's a responsibility of whosoever researcher is doing some sort of data analysis on top of that, or you have some sort of interface created which is even evaluating data collected out of that.
So I think if Arjun's still on the call, maybe he can answer that, because it's his dream really. That's a very good question, Bhupendra. It's his dream to start a biobank for this kind of data. We don't make the software for it, but everyone's got their own algorithms, because it's not like a one solution fits all model. So
That's correct.
people have been trying this, but Arjun, if you're on the call, maybe you'd like to take that.
I think Professor Arjun has left right now. I'm not sure if he's in the call.
Never mind.
I
I think.
okay. Right. So Bhupendra, yeah, we don't make them as yet. We don't make the software as yet, but there are certain companies like Intan and NeuroNexus who make these commercial packages. So there aren't a lot of them, and most researchers use theirs. But the whole point is it's not just about recording, but it's about decoding. So when they figure out the cause and effect relationship, that's what's important. So the human connectome project, right, that the world's announced, there are some open source websites on there, open source resources on there, which allow you to communicate and share this data. But we don't do it as of now. Arjun does it in his lab, but we don't do it commercially.
In fact, I have started some initiative around that. I'm from even research background, so I'm doing some sort of analysis on the algorithmic side. So I think I'll connect you offline. So let's see how we can actually utilize to this props in our further research perspective. And
yeah, absolutely.
And
so please feel free to drop an email or message me, and then
I just send you a LinkedIn connection, so we'll connect you offline.
Yeah.
So
sounds good.
Yeah. Okay, I think that's great. So the data, in fact, I mean, once you implant these props into the brain, right? So then what kind of data is actually generating? What kind of information you are providing? Decoding is something I understood.
Yeah. So we can record right down to single units. So like information, record action potentials from a single neuron. And some people prefer to record LFPs. Some prefer like other more superimposed signals, but we can go down to a neuronal level. So we can pick up the dimensions of our electrodes are 15 microns or less, and with those we can definitely allows for neuronal recordings. Ideally, typically, I think rule of thumb is the separation between neurons is about 80 microns. So having a 15 micron electrode placed between them is quite comes in quite handy to record that information.
You have some sample data collected out of that?
I do. I don't think I have it on the presentation though, because it's like ethical, legal things. Right? Like it's someone else's data. But I'd be happy to share it offline.
Yeah, let's connect offline. I think this is fantastic, and we're really proud. At least you started from India, and definitely this is something Southeast Asia. I mean, I'm basically working in the similar area. Maybe I'll share more offline. So I'm as of now based out Malaysia, but basically from India itself. So I think there are lots of potential in Southeast Asia market. So in fact, not Southeast Asia and entire Asia market, I would say, compared to US and Europe, and they are quite ahead with the Asia market. So there are definitely a huge potential. And most importantly, is something we are, you guys are doing for a community. It is definitely trying to solve some very, very critical problem, which is actually giving back to community.
That's true. Thank you. Thank you for the kind words, Bhupendra. And thanks for.
Hi, Kenny.
Hey, Vedant.
How are you?
Good.
So I had a comment on what you're discussing just right now. So I think not only for the side of recording, but also for the side of stimulation, there is a huge open market for providing stim engines, if you can say, and I just wanted to add on to what the other person was actually talking about, and probably investing in stim engines. And if you're looking at stimulation and therapeutical stimulation, for example, that opens up another market altogether. So is there a plan to develop that? And if you could remember, even in Neuralink, we were trying our best to come up with a solution, right? So so that's basically a comment and a question together.
I agree. I think the difference between Neuralink and Eva Neuro is we're not as big of a company. So for us, we plan on using OEMs, and then there are other companies out there who make stem engines and packages. So the whole idea is to be modular. So if we make a device that can interface with commercial off-the-shelf packages, medical packages, then that's the goal. I think Neuralink had its own monolithic package. So for them, it was a different challenge interfacing all the components in-house. But at least for me, the plan is to connect to other companies like Integer or Nuvectra and then use their engines. But it's still a little far out, I would say. Thanks for the question, Vedant.
Oh, you're welcome. Thank you, Kenny, for the wonderful presentation.
No problem. Thanks.
Thank you, Bidan. Any other questions?
Yeah, I wanted to say something. So yeah, super amazing talk. And so I'm Shrutika. I'm from Arjun's lab. I'm part of the decision making lab. Yeah. So the discussion was super amazing. I will convey this to Arjun as well. I think you would also. But yeah, I had the same. I'm an undergraduate student, and the kind of, this was my first talk in this domain, particularly because we study mostly decision making and mostly on the academic side of it. So the technological side. So I wanted to just say that it was really nice. It was on a level that even I could understand, I felt. And another question, I think it was covered somewhat, but when we talk about invasive, how invasive it is, I mean, on the aspects of pain, basically pain to the model organism. How would you, in what range of pain are we saying? You talked about and everything.
That's a very good question. So for humans, this is amazing, right? But just we feel pain in the rest of our body, but pain is communicated to the brain through pain receptors called prostaglandins. The brain doesn't have that. So you could poke someone in the brain with a knife and they would not feel anything because the brain doesn't have any pain receptors. So for whoever's undergoing the surgery, there's no anesthesia. It's an awake patient surgery, and then based on that's why, like, if you watch some neurosurgeries, the patient's always awake and there's feedback. There's someone in the front, someone pokes in the brain and says, "Is this the region we're into?" And then the person moves in there or something. So there is no pain because the brain doesn't have any pain receptors.
Wow,
that doesn't mean we implant too many of them because you'd cause a lot of damage in the brain without really feeling anything. But patient doesn't feel any pain. And for other animals, there's always anesthesia that's administered. And it's when you look at the scale, right? Like these are really tiny compared to our brains. So it's even like these are our blood vessels, right? So they're tinier than the width of the blood vessels. So it's unlikely that it's going to do a lot of damage. And you can see that the explanted probe also is like neat and clean, and you're not pulling out like part of the brain. It's fairly smooth, and it's a good process. Yeah.
So that's very exciting to know, because we are talking about invasive, but it's actually not that invasive now that you talk about it. So yeah, really exciting. I did not know about this aspect at all. So very nice.
I'm glad the talk. Thanks for joining in on a Saturday.
Thank you.
Sure. Any other questions from the audience? I think all of you were excited. Right. If there are no other questions, then we can just conclude the talk. Because already I and Kaustubh have had enough discussions on technical. So I think my queries are somewhat completed. So my only final question is, I mean, not a question, but I just wanted to know, where is Eva Neuro right now? What are the next 3 plans that you have?
We're still young, right? We've made prototypes and we've made some money by selling to research labs. Eventual goal is to chart the road for clinical translation. So we're because technology is not very different, right? Like, again, I don't mean to say this. I don't mean to be very like cocky saying this or egotistical saying this, but the technology in India or like Malaysia or the US, it's similar because it's the tools that are used to make these things. They're not humans, or they're not. It's not too far. It's not too different. So what Neuralink is making or what NeuroNexus makes. The same technology is available in a lot of places. Like any place that makes semiconductors, this technology is readily available. And I'm not even using technology that's recent in semiconductors. This 1 micron was made in the 1980s, right? For when the same technology that the Pentium processor used. So Pentium, I don't know if most people on this call are even as old to remember the Pentium processor, but it's an old technology. So the tools, but the utilization to make not just electrical but mechanical structures is what makes it novel. So the whole premise is that we, for AVA at least, we want to take it slow. We want to make this technology readily available. That's the first milestone. But after that, at least we expect in Q3 or Q4 of 2022, where we've applied to a couple of academic grants. If we get them, we want to chart the road to clinical, where we develop not these designs, but like one design that might have clinical translation, and then continue on with animal studies. So the animal studies themselves go on for a couple of years. And if the data, these are called GLP, good lab practice studies. So if they're collected, and once we get the regulatory approvals from ISO and FDA, it's only then that we qualify for human trials. So it's a long journey, but we're still young, so I don't mind. Hopefully by the time I get Parkinson's, I want this available, because I don't want to drop my glass because of the disease. But it's, you got to push, right? Like after the work that people did in the 20th century was available to us. Our work we do is available to the generations after. So that's the goal. But the dream is to have a clinical device, and that's where we start.
Great. I mean, I'm able to feel the enthusiasm and excitement that you have, and all of us share, right? And as one of the people in the audience said, it's very proud for all of us that our Indian startup is making really highly advanced neural probes and is competing with companies like Neuralink. That's a very proud moment for us and for the whole ecosystem. First of all, I need to thank you and Arjun for that. Thank you so much, and hope we all are there to support you, because currently I've been recently pitching with neurologists and neurosurgeons about the community. And even this afternoon, I had pitched the community to NIMHANS neurologists. So we are always there to support you in any case if you want to do any connections with Indian neurologists and neurosurgeons, right? We're always there, and it's a proud moment for all of us, and it's our responsibility to do that for the startup. So yeah, please feel free to reach out to the community, and I've also shared the community links. If somebody's any of you is interested in join Neurotech X India, you can mail to india@neurotechx.com and also follow our social updates. And finally, I was very, very excited. This is the second time I'm hearing all the technical information. I'm still excited, and thank you so much, Kaustubh, again. And all of us in the community, we feel the same sense of passion. And I think when he was explaining about the brain and getting into the brain with the probes, it was complete magic. And we feel that we are the new generation magicians who are dealing with the brain, and we fall in love with the brain, right? That's super exciting. And hopefully, this community gets together, all of us gets together, hug the brains, and just engineer with it and do something really impactful for the whole world. Yeah.
Thanks, Karthik. Thanks for the kind words. And I just want to add to that, as humans, right? We figured out what this monk does. We figured out what the legs do or what the arms do. The brain still evades us. We're in the 21st century. We've mapped every tiny corner of the earth, but we still don't know what our brains do before. We know a little bit. We know what certain areas of the brains do, but we can't treat them. And I feel as evolution progresses, it's unlikely that we're going to go from Homo sapiens to something else. It's going to take a long time. But if we get to that stage of the android, figure out the brain, man, that'll be so magical. All those dreams about, can you fly? Can you, jump as high as 3 floors? Or can you, even basic things like losing weight or control, your urination. All those are possible with stimulation, because oftentimes you realize, and especially a lot of people who've got these injuries tell me that come to know that our arms, our limbs, everything else, it's just mass. It just matter. There is nothing more to it. It's the brain and the spinal cord, and of course the heart, that makes us what we are. Because the moment your brain stops functioning, any of the nerves in your arm stop functioning, the rest of the arm is just matter. I mean, alternatively, if you're able to use a prosthetic arm or an exoskeleton or even a jetpack for, I'm throwing out ideas, like audacious ideas, but it's possible. And we're starting out, like we're still at 1%, maybe 2%, but it's possible. Like we could do it.
Definitely, like that's the future is super exciting, and that's why, right, all of us.
I just want to read out Tom's comment here. He says, if the brain was simple enough for us to understand, we would be too simple to actually. It would be too simple for us to actually understand it. That's correct. That's correct.
Right? Yeah.
Famous
Thanks,
word.
Tom.
It's a beautiful quote. There has to be some magic to life. I agree, but I'm sure by the time like we cross over to the next evolution, I'm sure there'll be some other magic. But yeah, exciting stuff, right? Like
thank you so much. And I think the first time we also had some clinicians over here. Like that's very exciting. And the whole mission of the community is to get all of them into one single platform. Like the professors are interested in India, and now clinicians are getting together. So we can get all of them into a platform and sort of rapidly progress the field of neurotech in India. So this one startup can open a way to a lot of other startups, a lot of other academic researchers in neurotech. And hopefully as an Indian ecosystem, we can definitely do it and be one of the forefront leaders in neurotech. Yeah, that's very exciting.
By the way, thank you very much, Karthik, for organizing such a wonderful event.
Yeah.
Thank you, Vipendra. Sure, and hopefully we'll have a lot of interesting events. Just follow Meetup and you'll be getting updates on, we're planning to have like 1 in 2 weeks events, and already the next week's the next speaker is also about to get confirmed. So that'll be an interesting speaker from UCLA, I hope. So yeah, hopefully, yeah, we'll have a wonderful time at Neurotech X India, for sure.
Awesome. That's great. That's great. Thank you, Deepak, for your kind comments. And also, just a moment of thanks for Neurotech X India, Neurotech globally. Tom, I'm sure like it must be an odd time of the day for you, but you still attended. So thanks for doing that. Everyone else on the call, like have a great weekend, and thanks for joining in.
By the way, I'm not sure, just last comment. By the way, I'm not sure if you guys know, there is a Slack channel, right, on a Neurotech. I think that is that's quite amazing Slack channel. So if you wish to join, you can get lots of interesting information there.
Definitely. I think you can see that on the Neurotech X website. You can just log into Slack channel. And if you log into Slack channel, you'll see #India channel. So definitely I would recommend all of you to join that to get updates, and we can have some discussions within the India channel. So every chapter, every country has their own channels. So yeah, that'll be very interesting. And thank you so much for that information, Mr. Bhupendra Mishra. Thank you. Yeah, hopefully we'll have a lot of good time together. And we also have a lot of events and webinars planned, like getting a plan to get all the whole Indian ecosystem onto one webinar, professors, clinicians, students, engineers. And that's one plan. And also to get global speakers. There's one more event is going to be planned. So hopefully we're going to have a lot of wonderful time spending with our brains.
Yeah.
Awesome.
Thank you so much.
Thank you all.
Thank you all. Thank you. Thank you.
Bye now.
Bye, Tom. Yeah. Thank you. Bye, all of you.